Hello, my name is Mark Case from the Science and Motion program at Lehigh Carbon Community College. Today, I would like to show you how you can actually use a subscription program that we have purchased called Graphical Analysis Pro. And this is from Vernier software. And what we will be able to do is to take real data, have the students view the experiments that are being performed, and then take their own computer or device and do analysis of the results of that particular experiment. Right now, there are approximately 40 different experiments in chemistry, biology, physics, middle school science, and you can choose any one of these that you can work with your students to see the results of an experimentation. Our focus today is from the Chemistry with Vernier experiment number 9, which is talking about evaporation and intermolecular attractions. Initially, what happens on the computer screen for the teacher is that they're going to see the first run. I can come up here to the top with this reverse arrow and reset this so that when I click on the play, what it's going to do is it's going to show me the results of ethanol versus 1-propanol. There's also a run that uses 1-butanol and pentane, or methanol and N-hexane, or a straight chain hexane structure. So if I start running this, you can also see that the speed that we have this being replayed is at 4 times the normal speed. So if the gentleman looked like he's going in there pretty quickly, yes. Number 1 is ethanol, and number 2 is the 1-propanol. We can see that they show up as 2 different colors. To help us correlate those, on the left-hand side, it says that temperature 1 matches up to this dark purple blue color that you have, and temperature number 2 matches up to the yellowish green line. Remember, number 1 was the ethanol, and number 2 was the 1-propanol. Down here under the graph tools, we have another option where we can add a legend onto this. When I click on here, it'll just show you the legend in the upper right-hand corner, and that'll help you figure out the changes that are occurring there as well. Now, what you can also do during this experiment is have your students be able to see this and view what's going on as you talk about the results. So, in order for you to be able to get that experience, what I have is my iPhone hooked up, and the iPhone is connected to my computer so you can see it at the same time. And if I pick on the graphical analysis app and turn that on, you will see basically the same starting setup that we had previously. What I would like to do here is to do data sharing. So going over and hovering on the data sharing and then picking that, it will now allow me to connect over to the computer that was being the teacher display computer. Now, there's a little broadcast symbol up here in the upper right-hand corner. When you click on that, that says data sharing, and you want to start your session, it'll generate an ID that you could use, and any device that's on the same connection using this ID on the internet will be able to see what's going on the teacher's computer. So the code is JZE 3GA for this particular session. And when I enter that, I have now linked up to it on my iPhone. As I mentioned, the iPhone is something that you could use with graphical analysis. It also works for any iOS device. It works for Android devices, so it could be an Android phone. It works on Chromebooks, PCs, and Macs. Any one of those will be useful for you to have that with your students. So when I start this over again, what will happen is that that data has already been downloaded to the student computer. So they have that data. If you launch the students at device before you start collecting data, sometimes they'll see a live readout of those results that are progressing as they move across there. On the individual data sets, what you can do is you can also display the other data. So the student could actually say that I'm not going to wait for the teacher. I can come over here and see that there's another trial using 1-butanol versus pentane. So if they click on that and turn that one on, they're now looking at a different data set as compared to what the teacher's trying to demonstrate. Then if you notice, there's a third one that's on there, which is methanol versus the hexane. They can see how those results will vary. So the teacher has those same um, different runs on their set, and they may take those on or off as they would so choose. For now, what I'm going to do is to make sure that we're both just looking at the ethanol and the propanol for your individual setup. You got an extra one on there, so turn that one off. Now, one of the things in this lab experiment that I asked them to do is to figure out the overall change in temperature. So the teacher could go down here and use the graph tools and say, show me the statistics for your results, your view statistics. And this will give you what your changes in time. We did 240 seconds for data collection. But what we'll see is there's a delta Y, the change in temperature, and we had 9.063 degrees for temperature number 1, which was looking at the ethanol. And then we have the one that is the yellow color, temperature number 2, the yellow green, and that's the one that we're looking at. It only went down by 4.483 degrees Celsius, and that's the one that was the 1-propanol for your individual setup. Now, this statistics that I did on my teacher computer does not show up on the student device. They would have to go and do their own statistics, turn that on by using the graph tools down the bottom. They should be getting the same answers here as they look at the data, because basically we're just doing statistics of the entire um, trials that we had for the ethanol and the 1-propanol. There's another feature that sometimes I like to look at to compare the rate at which they were evaporating, and that is if I highlight the portion of the curve where the lines stay fairly similar, fairly linear, and they don't get to a point where they start to bend off of that straight line, I can come down here under the tools bar again that I have for the graphs, and I could do that. I want to add a curve fit. Now, the curve fit that I want to apply is going to be linear, but I would have other tools that I could use if I so chose to look at some other mathematical relationship that might fit. So when I go apply, and again, I can drag this bar up or down and I can minimize it and so on. But what this is showing me is that the slope of the temperature 1, which was my ethanol, is an M value in the Y equals mx plus b of negative 0.12. I can carry that further, but sig figs, that's good enough. So it went down by 0.12 degrees per second is what that comes out to be, because it was delta Celsius temperature versus the time. If I look at the yellow green color one, temperature number 2, which was my 1-propanol, that slope is negative 0.0334 if I round that up. So it was changing much less than what we were getting in terms of taking a look at the ethanol. Again, the student would have to come over to their own device, tap on the graph tools, choose the curve fit, and now the curve fit that I am going to be using, I would have to highlight the portion that I want to look at. So we don't want the initial starting temperature. If I go too far, I can grab one end and I can shrink it down a little bit. Or if I went too far on the other side, I can bring it back a little bit. But now I do my curve fit, my linear fit, and their slopes might be slightly different than the ones that I had. So I ended up with a negative 0.112 and a negative 0.034, but depends on where I would say that the linear curve started to lose its fit. At this point, the student could do any analysis of the other liquids that were involved. So they're pretty much on their own. They could do the same thing with the butanol versus the pentane, look at the differences. If they tap anywhere along the curve, they can read what the values are. I, as the teacher, could be doing that same thing, but it would be good if they could explore on their own and try to see if they can derive some interpretations of the data. The other thing that they can do is when they are done, they can go to the untitled up in the upper left-hand corner, and they can either save this. Usually it's good to save as and give a name to it, and save it to a folder where they might come back to it later on. There's also another option where you can do an export, and you can either export it as a CSV file, which is an Excel type of spreadsheet file, or what's called an AMBL file. That AMBL just means that it was created with graphical analysis as a suffix that goes along with that. So they could do that either way. A third option, which is helpful for teachers who want to be able to see their results, is most kids are savvy enough to be able to take a screenshot. And once they do that, they can edit it. It'll go to their picture files, their phone files that they might have, and then they can do the editing from there and trim it down and make it look nicer and so forth, and cut and paste as usually a PNG file or a JPEG file. So that would be how you collect and store data. That was just one short look at it. In the second part of this video, what I'm going to do is show you how you can actually access um, other data that you don't have in this Graphical Analysis Pro version yet. It's not up and running, but you can still use data and analyze it from there. The third thing that I want to point out before I go to the part two is that when you examine and take a look at each one of these individual labs, there is a Word file that is editable, so you can actually change this and adjust it. This is the Chemistry with Vernier, experiment number 9, and it gives the background of what they're trying to do as they're studying, looking at alkanes and trying to compare those to alcohols and see what differences they will have in intermolecular forces. It's showing you the general setup of how you're doing the experiment and using the Go Direct temperature probe to be able to collect your data, what materials you would need to be able to go through it in this fashion as they did here. And then talking about the types of intermolecular forces, the fact that you will have, excuse me, dispersion forces of the nonpolar substances where the electrons are going to repel each other and build up a temporary dipole. But then you also might have some hydrogen bonding when the hydrogen is bound onto a nitrogen oxygen or fluorine. So it goes through the process, it gives you the procedure, and then probably more significantly down here, it directs their thought process in terms of how do you process that data. So this information, how do you process it? Where do you get the teacher notes so that you don't have to try to look all this stuff up yourself and you know what to expect for that? That's in the next video. So we'll see you then.